So we started going through this review sheet yesterday. Before I dive in, are there any specific questions you guys have at the moment? I don't think anybody's brain's firing at full power today, but we'll do our best. Okay, so we went to the first page. After a couple of these, right? We were talking about enzymes, yeah? We were talking about we were talking about like pH and stuff like that, right? Okay. So we know the purpose of enzymes is to speed up chemical reactions through lowering activation energy, right? And we were talking about last class how things like pH and temperature and salt concentration can cause the denaturing of a protein, correct? Okay, so let's just go through these, make sure we're good. So that you can explain how the cellular environment affects enzyme activity. You've got to be the correct temperature, pH, etc. You can explain the difference. Hydrogen ion concentration and varying pH. Where do you have more hydrogens? At what pH? Lower. So just remembering that it's inverse. You can explain the way the concentration of reactants affects enzyme activity. Why does greater concentration of reactant equal greater enzyme activity? It increases the chance of what? Collision. Remember, it's purely by collision. So if you increase the number of enzymes, increase the number of reactants, or both, you're going to increase your overall reactant rate. This one's an interesting one. I can explain the concentration of products effect on enzyme activity. What do you think? Do the amount of products have any impact on enzyme activity? What are you thinking? No, okay. Any thoughts? Right, and you're exactly right there. Tom, did you want to build on that?
No, I just want to say, like, if there was an equilibrium reaction, like products, like if we added products, it would shift to the.
Sure, if you want to look at it, which you're correct, chemistry-wise, that definitely would happen. And that's not incorrect by any means, but it is looking at it more from how others are saying it, in that as we have a lot more product, you're indicating we have less reactant, but also look at it from the reactant has to find the enzyme via collisions. If we have a lot of products floating around, it's going to be less likely that the reactant can find by random collision the enzyme versus the product, because there's so much product floating around. So it's not that the product really has any technical impact besides making collisions less likely. Good. Okay, temperature we talked about. Hot denatures, what does cold do? Just slows it down or stops it. Good. Where does a competitive inhibitor work? In the actual active site. Very good. What could I put in to overcome a competitive inhibitor? What could I add to an already existing reaction? Very good, because if I add a whole bunch more reactants, then it's more likely that the reactant happens to collide with the enzyme than the competitive inhibitor reacting with the enzyme. Excellent. What's a non-competitive inhibitor more often called? Or what's the other word we can use for non-competitive? Starts with an A. They're also allosteric, because non-competitive just means it's not fitting into the active site, which is the same thing as an allosteric inhibitor. They work anywhere but the active site. So how does an allosteric or a non-competitive inhibitor inhibit? How does bonding to somewhere else on the enzyme make it stop working? Exactly. So when it bonds somewhere else, we change the shape, which changes the function. Excellent. And you made a perfect note that I want to bring up. If you're doing this in a short answer or an essay format, make sure when you're talking about changes to shape or changes to structure equal changes to function, that you specifically note it's the active site region of the enzyme that has a different structure. Because theoretically, you could have a change that leaves the active site intact and only changes a different portion of the protein, right? So, and that wouldn't have any negative impact on the way the enzyme works. So if it's on an essay question, just always note that it's the active site structure that's changing, like you did. The pH formula is technically on your formula sheet. I've never once seen them make you use it. It's basically, you just need to understand acids have greater hydrogen ion concentration. Okay? Any questions on that? Okay. So then this is what we were talking about with energy. For our purposes with energy, you need to be able to understand that endergonic and exergonic graphs. Know where the reactants are, know where the products are, understand the activation energy, understand what an enzyme being added to the graph would look like. You could draw or interpret those graphs. Questions on those? Okay. Can you describe what energy coupling is? Yep. Using the exergonic reaction to power the endergonic reaction. Can you describe loss of energy as energy flows? That's what we talked about with, like, when we remembered the trophic levels. We lose energy as what every time we transition it? Heat. Very good. All right. We'll keep going. Finally getting into photosynthesis. So with photosynthesis, you want to be able to take your diagram, which, do any of you guys still have diagrams in the back? If you do, I would take them home today to study from, ideally. Okay? But you need to be able to fully explain that diagram to someone else. If you could get to the point where you can teach that to someone else, you are at a very solid place with photosynthesis and cellular respiration. Let's look at these. I can describe the photosynthetic process. That whole point is just the overarching, converting solar energy into chemical bond energy. I can describe the location of photosynthesis evolution. That's talking about how it started out in prokaryotes, and then like the endosymbiotic theory, where we think the chloroplast was like originally its own prokaryote that got engulfed. I can provide evidence to support oxygenation of the atmosphere. You don't need to be able to provide evidence. We're going to do those early life hypotheses when we get to evolution, but just knowing that photosynthetic organisms are who oxygenated Earth, that's the hypothesis that's out there, the theory. All right, I can describe the light-dependent reactions. So, can you tell me, where did the electrons originally come from for the ETC? Where do they end? Where do the hydrogen ions come from to create the concentration gradient? How do we replace the missing electrons, right? If you can answer all those questions, you understand the light-dependent reactions at a good level. I can describe the ways that ATP is synthesized, so understanding we purposely create a concentration gradient to allow it to flow through the ATP synthase. That process is technically called chemiosmosis. I know you've seen the term in your book. That is the technical word for ATP synthase, how ATP synthase works. You know what osmosis is. Chemie is chemical, so it's chemical osmosis. It's the diffusion of chemicals rather than water across a membrane. That's what it stands for. I can describe the ways that NADPH is synthesized. Yes, you should know that that's our final electron acceptor is NADP+, and then it grabs hydrogen ions that are also floating around. If you have questions as I'm going, please make sure to ask them. I can explain the way cells capture energy from light. So that's our pigments. The one we talked about most often was chlorophyll. Understanding that these pigments are special molecules whose electrons can be excited by solar energy. Not all molecules can do that, right? I can explain the way cells transfer energy from light to biological molecules for storage and use. That's the whole light-dependent, light-independent reactions. That's basically the whole thing, that the point is we turn it into ATP and then use the ATP that's produced to power the endergonic reaction of building glucose, which is or carbohydrates, which is the energy storage unit for the light. You can describe chlorophyll. You understand that it's a light-dependent pigment. It's able to have its electrons excited by light. I can explain the effect of on electrons after absorbing energy from light. That's that they gain energy of position, so they move away, right, from the nucleus of the atom they're attached to. They become excited. I can describe the anatomy of the thylakoid membrane. You can describe the anatomy of a chloroplast in general, thylakoids, granum, stroma, the two membranes. I can describe a photosystem, that it's a transmembrane protein that has the pigments embedded into it. I can describe the relationship between the photosystems and the electron transport chain. The photosystems contain the pigments that get excited by light, which allows the electrons into the electron transport chain to power the proton pump to create the gradient so that it can flow passively through ATP synthase. I can explain ways the proton gradient is produced. We know the hydrogen ions originate from water and that they are pumped into the thylakoid space. Here's a good point too I want to bring up. How do scientists know? How do they know the hydrogen ions get pumped into the thylakoid space? How do they know they get pumped into the inner membrane space of the mitochondria? Like, how do they actually know which direction all these proteins are facing? They actually do it by watching pH and testing pH, because hydrogen ions, pH is just power of hydrogen, right? So they can test pH levels inside the thylakoids versus outside, or inside the matrix versus outside at various points in cellular respiration and photosynthesis. And they know that when the pH drops lower, that indicates what? More hydrogen ions, which indicates that's where the gradient's being created from. I can explain how the proton gradient leads to ATP synthesis. That's because it flows through ATP synthase, and then the bulb portion spins. And I can explain the relationship between light reactions and Calvin cycle. Light reactions create ATP, which is necessary to power the endergonic reactions of the Calvin cycle. Questions? Good so far? Now. Cellular respiration. Cellular respiration, while harder in a lot of aspects, should be easier if you've got a good handle on photosynthesis. Basically, you understand that solar energy has been stored as potential energy within the bonds of the glucose molecule when it's in the carbohydrate molecule. And then we're going to spend all of cellular respiration systematically breaking apart the carbohydrate into its component pieces, collecting the electrons, collecting the hydrogens, and throwing everything else away to then power an electron transport chain so we can get hydrogens to flow through ATP synthase. Okay? I can describe the process that allows organisms, that's why it's mumbled. I can describe fermentation. Fermentation is when there's usually oxygen available as that final electron acceptor, but we're currently under a condition where it is not. So the organism does glycolysis only and produces a carbon-based molecule byproduct to allow them to make at least a little bit of ATP while there's no oxygen available. Be careful you don't accidentally write me an essay that indicates humans can live without oxygen. Our skeletal muscles can do fermentation. The rest of you cannot. Okay, so don't indicate we die without oxygen. Don't try it. I can describe the electron transport chain. Same thing as photosynthesis. Can you describe where the electrons come from? Can you describe where they end up? Can you say where the hydrogen ions come from? Can you explain where we get our gradient from? Can you identify where it happens? That's on the cristae membrane. You can describe the final electron acceptor. That's going to be oxygen in aerobic respiration, and it's going to be something that's not oxygen in anaerobic respiration. I can describe the effect of high proton concentration on the pH. That's what I was talking about, of how they actually dictate it, which way all of these proteins are facing and how stuff's actually getting pumped and flowing. We can trace that by tracing the pH. Occasionally, they will ask a question that references pH instead of hydrogen ion concentration, but be aware they're the same thing. I can compare the electron transport chain in eukaryotes and prokaryotes. We don't get into that much, okay? The only difference is, no, because they don't have organelles, their electron transport chain happens on the actual cell membrane rather than like an interior membrane, because they don't have interior membranes. Chemiosmosis is the technical name for the protons flowing through ATP synthase. Oxidative phosphorylation is the ATP we make from ATP synthase. The ATP we made in glycolysis and Krebs cycle is called substrate level phosphorylation. Oxidative phosphorylation is way, 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 way more efficient. Don't worry about photophosphorylation. We're not doing that. I can describe the ways that endotherms maintain body temperature. This is just an example. You guys correctly told me every time we transfer energy, we lose it as heat. So endotherms, or organisms that maintain internal body temperature, will do this on purpose in situations where their body temperature is too low. Why do we shiver? When we're cold, you're purposely generating heat. You're purposely doing chemical reactions that transfer energy, knowing some of it's going to be lost as heat to help warm you up internally. Okay. Questions on those? No, I've never specifically seen them ask that, but if they were to give you a diagram that had the numbers on it, you can make sense of it. Does that make sense? Good. Yep. No, I would not waste time memorizing the numbers. I don't think that's particularly useful in terms of like memory space. But if they give you a big old diagram, you could follow it through and understand, oh, this one came from here, this came from here, you know, and where we end up. You should know the formula. That is important to know. Other questions right now? Last page 51. Okay, we went through a lot of this already, but I can explain ways cells obtain energy from biological molecules in order to power functions. The primary molecule in us as humans is obviously glucose. Any carbohydrate gets converted back into glucose. But we did note, yes, other things can be used. Lipids just get converted back into carbohydrates before they're used. We have to be in a starvation situation to use proteins and nucleic acids, but we can. But then obviously things like carnivores, they have a slightly different system to just use the lipids and then proteins if they have to. So just depends on the organism you're talking about. But if there's ever a starvation situation, your body can break down any of the 4 macromolecules to do some form of cellular respiration if they have to. Glycolysis, take the glucose, break it in half into pyruvates. We make our first NADH. We make some ATP. Starting materials of glycolysis are glucose, but also technically ADP and NAD+, right? Because we need those to make the ATP and the NADH. Where the parts of cellular respiration take place, the big key there is that glycolysis does not happen in the mitochondria, and that's like a universal process. The Krebs cycle and pyruvate oxidation happen in the matrix. The electron transport chain happens on the cristae membrane. I can describe the Krebs cycle. That's basically the inverse of the Calvin cycle. We're going to pull everything off. We're going to pull the remaining carbon dioxides off. We're going to pull the remaining hydrogens off, the remaining electrons off, and that's exergonics. We're going to make a little bit of ATP. Starting materials of Krebs cycle, acetyl-CoA goes in, NAD+ goes in, FAD goes in, ADP goes in. Products of Krebs cycle, ATP, FADH2, NADH. The results of the electron transport chain is that energy is produced, which is used to power the proton pumps, which force the hydrogen ions into a location, which allow them to passively flow back through the ATP synthase. The function of fermentation is to keep the organism alive when there's no oxygen available to be the final electron acceptor. ATP hydrolysis, the whole point of why we're making ATP is then when you break ATP, it's very, very exergonic, and we use that to power everything else the system's doing. Anything on that? And then the last one, fitness. I told you to not overly worry about this, but this is the CAM and C4 plants we talked about. So don't even worry specifically about these questions, but it's just a note of that evolution fits into every single topic we're going to go across, we're going to discuss in this class. Why are things the way they are? It's because this is the point they've evolved to. It's where we're currently at. But things like, how do we have plants in a region that don't support plants because they lose too much water? Well, we've evolved C4 and CAM plants that take photosynthesis a little bit differently, right? It's just understanding that concept. You don't need specifics about anything. All right. What else can I answer for you? Anything? You in Thanksgiving mode already? Yeah. Okay.
So I remember you saying that when it takes energy to build bonds, it creates energy when you're building bonds, right?
Correct.
And it takes energy
To break a bond. Yep.
But then also to go from a simple structure to a more complex one, it all fits endergonic, so it takes energy to do that.
Even though it seems like you're mostly building and it should be exergonic.
Yeah.
Yeah, that, let me go ahead and explain that a little bit. So to reiterate, because I don't know that people could hear you, to build a bond individually releases energy because it's coming together and they're moving to a lower energy state. To break a bond, you have to put energy in because we're forcing the electrons back to where they're supposed to be, right? But when we talk about like, okay, when you take simpler molecules and build them up into a more complex molecule, it's going to be endergonic. But if you're looking at it and going, it seems like I'm building more than I'm breaking. It's just the way it appears. If we actually went through and did the math, and you can look up bond energies for each individual bond, and we could say we're breaking this whole set of bonds, we're building this whole set of bonds. It does work out to in net require an energy input. It's just deceiving from the outside. It definitely looks like we're going against what we said. But if you go through and actually do the whole summary, like what we're talking about with, like, say, photosynthesis, simplistic and more complex is going to be endergonic.
And then more complex
Will be exergonic. And same thing, even though it appears like we're doing more breaking than building, if you go through and actually calculate all the bond energies, it's not. Yeah, it's just one of those where it's way more than you realize. You look at it and you're like, that's not that many bonds. But if we actually go through and do it, did you guys have to do that in GT Bio? Do you remember the pink packet where we made you actually calculate all the bond energies for cellular respiration? Getting like a couple nods. Most people hated us, and you might have like permanently blocked it from your memory. It was definitely in the pink packet, and we gave you like the bond energies for like a C to H, an O to H, an O to O, a C to C. Right now I'm getting some angry nods. And you actually had to calculate like, okay, we built this many waters, we broke this many oxygens, yeah. So if you actually do all the math, it does work out. It just doesn't always seem that way from the outside. But we are not breaking any chemistry rules or thermodynamic rules, I guess. I'm not going to make you do that again. You guys whined through it the first time, but I promise it works out. Anything else?
So for like the
Right.
we just have to like see where that
Correct. There's no way for you to look at an enzyme and know what the optimal range is. They'd have to tell you or show you a graph or do something, and you know if it just goes either direction out of that optimal, it's going to cause problems because basically they have to fold up into a specific shape. We know that's based on the R groups, right? But it's also based on the pH of the surroundings and the temperature of the surroundings. And they're designed to fold up into a specific shape for that pH, for that temperature, for that salt concentration. So when you move it, even though you didn't change the R groups, you're technically going to get a shift in shape, even if it's minor, and that is going to shift the function. Because there are enzymes that work at all sorts of pHs and temperatures and everything. It's just whatever is that enzyme's optimal. Like, they have found enzymes that can still function in above temperatures of like boiling, right? They used to think boiling was like perfect sterilization. It killed everything. It doesn't. There are organisms that can live in boiling water. So their enzymes work. But if you were to take that enzyme and cool it down, it still doesn't work as well. Or if you take it above even higher temperatures, it's still going to denature. But it just works perfectly in that temperature. Same with pH. You guys have stomach enzymes that work in a pH of 2. You move them to a pH of 7, and like your blood, they denature because they're designed to exist in a pH of 2. So the moment you add way less hydrogen ions, they actually clump up on themselves and don't work. Anything else?